morning, dear students. Today we are going to continue the balance portion of mitochondria chapter. And in the last class, we have learned till mitochondrial oxidation. Today we are starting with the oxidative phosphorylation. What is this oxidative phosphorylation? It is simply the synthesis of or generation of ATP from ADP as a result of a series of oxidation reactions. So many oxidation reactions are happening, and as a result of that, ATP is forming from ADP. That is oxidative phosphorylation. It's a very important question. There is more chance for having this question for your university examination. So study well. Don't omit this question. Next is chemiosmotic hypothesis. Also very important. What is chemiosmotic hypothesis? Who proposed this? Peter Michel in 1961. What is this hypothesis? It is a hypothesis. Guess it's a theory based on speculation, guesswork. What is that? This is mitochondrial inner membrane. And in this inner membrane, we can see so many enzyme complexes. And through these complexes, electrons are moving. From where those electrons are coming? Those electrons are coming from some electron carriers. That electron carriers carry that electrons, or they take that electrons from various biogeochemical cycles that happens or undergoing inside the mitochondrial matrix. And from those cycles, they got that electrons and they carry that electrons to these complexes, and through these complexes, the electron pass in this direction, pass one complex after another in that way. Okay, that's what I am showing here. And because of this transport, enormous amount of energy is liberated. And using that energy, what is happening? Protons from mitochondrial matrix, this is matrix is transported across the mitochondrial inner membrane towards the intermitochondrial space, means the space between outer membrane and inner membrane. Okay, I will show that this is inner membrane and there is outer membrane, and this space is called the intermembrane space. Intermembrane space, and towards this space protons are transported. And as this space is very narrow, very easily we can create a high concentration of protons here. Okay, there's a process, and as this process continues, very soon itself the concentration of protons become more in this space, and as a result of that, proton can very easily move again towards the mitochondrial matrix side through another protein complex called the ATP synthase. I'll show once more that proton, as its concentration is more here, it passed through another enzyme complex, and because of that transport, that movement, as it is a downhill movement means a movement from a region of higher concentration towards a region of lower concentration. Because we know that, we understand that proton's concentration here is now very high, and because of that high concentration, proton can easily move towards the matrix, and that is a downhill transport that also can liberate enormous amount of energy. And using that energy, the ATP that is formed here inside this ATP synthase enzyme. The enzyme itself cannot release the formed ATP, and it released that formed ATP by utilizing the energy released as a result of the downhill transport of proton. Okay. Okay, then what is chemiosmotic hypothesis? Hypothesis is that this electron transport, it is coupled with the phosphorylation and synthesis of ATP. So this is very simple. Simply in this way we can give the answer for the question. What is chemiosmotic hypothesis? Proposer is this scientist, and what is it is the coupling of electron transport with the phosphorylation of ATP. Next. Next, we are moving to the electron transport chain. And in the electron transport chain, the main process, I'm going to explain you about the important points and process that is happening in the electron transport chain. First point is that two energy rich molecules they are NADH and FADH2, means these two are electron carriers. They got their electrons from different cycles, biogeochemical cycles that is happening inside the mitochondria. Okay, then they are in reduced form. These two high energy rich electron carriers are oxidized. They got oxidized in the electron transport chain. This is a one important process that is happening in electron transport chain. Then next one, this electron transport chain involves many oxidation reduction reactions. Next is redox couples. Redox couples means in the electron transport chain, so many ions are present, so many compounds are present, and they undergoes oxidation and reduction. Um, so that there are two forms, oxidized form and sorry, and reduced form. These two forms, its pair is called redox couple. And next is oxidation reduction potential. Potential means its ability. Ability for what? Ability to transfer electron to an acceptor. Means it indicates the ability of a substance to transfer an electron to an acceptor compound. That is oxidation reduction potential. Then respiratory chain. This image also you can refer. Respiratory chain, it consists of a series of electron carriers. Electron carriers, example NADH and FADH2. They are arranged in the order of redox potential. Redox potential means their potential to get reduced or oxidized. It is high for NADH, so that's why we can see NADH first in this series, and later only FADH is coming. That means FADH have less redox potential when compared with NADH. Then enzyme complexes. Enzyme complexes means these all are enzyme complexes involved in electron transport chain. And if NADH acts as electron donor, what will happen? This is complex 1. Then the electron from the NADH pass through the complex 1. That passage of electron happens across the complex 1 only in which situation? If NADH is the electron donor, then only in that situation the electron pass through the complex 1. This is a complex 1. Okay. If FADH2 is the electron carrier, then what will happen? This is FADH2. FADH2 is the electron carrier, then electron moves through the complex 2. Then another point we need to understand is the downhill transport of electrons. That means electrons are transporting in this direction. It will release enormous amount of free energy, and using that energy, protons are translocated from metric site to the intermembrane space. Okay, in that way, a proton gradient, concentration gradient is developed in the intermembrane space, and using that gradient, ATP is forming. So that's what explaining here in this slide. Next, we are going to see about each complexes. What are the processes happening in each complexes in detail. Okay. First complex is NADH dehydrogenase complex, or otherwise simply complex 1. What was going on here? In the former slide, I have told you that if NADH is the electron carrier, that electron will pass through complex 1. So that's happening here from the TCA cycle, that is citric acid cycle. Electrons are generated. That electrons are picked up by electron carrier NADH, and it is reduced, and from that reduced NADH, 2 electrons and 2 protons are entering this complex 1. It is passing through FMN. FMN is a cofactor, and from the FMN again the 2 electrons and 2 protons are moving to another electron carrier called ubiquinon. When ubiquinon got these electrons, it attained the reduced form, that is the ubiquinon form. Its oxidized form is called the ubiquinol. Okay. Another important process that is happening here is the translocation of protons from mitochondrial matrix towards the intermembrane space. Okay. Next, we can move to next complex. This is the second complex. It is called succinate dehydrogenase. And this complex is having 4 subunits. In the citric acid cycle that happens inside the mitochondrial matrix, there is a step that involves the conversion of succinate to fumarate. During that step, 2 electrons and 2 protons are released, and those electrons and protons are received by an electron carrier called FA D, and it get reduced to FADH2, and from this FADH2, that electrons is moving to the complex 2. Inside the complex 2, an iron sulfur center is present. It after receiving these electrons, this also get reduced and from this reduced iron sulfur center, that electrons and proton move to ubiquinol and reduce it to ubiquinone form. But here there is no free energy is generated. Because of that reason, there is no translocation of proton from matrix side to intermembrane space. That is another important thing that happened with respect to complex 2. Okay. Next complex is ubiquinone cytochrome oxidoreductase. And look its name, ubiquinone cytochrome oxidoreductase. Ubiquinone, we know it is the reduced form, and the cytochrome, first it stays as oxidized, and in this process happening with in connection with this complex, ubiquinone undergoes oxidation and cytochrome undergoes reduction. So that's why this complex is called the ubiquinone cytochrome oxidoreductase. Then we can see what is the process that is happening associated with this complex. We need to begin with this 2 ubiquinones, reduced ubiquinones. This ubiquinone gain the electrons from complex 1, and this one gain that electron from the complex 2. And from these 2 reduced ubiquinones, 2 electrons each is moving to cytochrome C. And that cytochrome get reduced. And along with that, Protons also is translocating from matrix site to intermembrane space and adding to the concentration gradient for this proton. Okay, next we are moving to the last complex, that is the complex 4. It is known as cytochrome oxidase. What's the name? Cytochrome oxidase. And the electrons that is carried by reduced cytochrome C is finally transferred to oxygen, and oxygen get reduced, and it is converted to water. Water, how it become water? After combining with the proton that is taken from the matrix of the mitochondria. So in one transport, two water molecules are forming, and that water is releasing from each cell, and a small portion is losing from our body in the form of sweat also. Sweat. Okay. So that's the last complex. And what's the reaction that is happening? Before that, I need to mention you that this complex is composed of 13 polypeptides, and its molecular mass is 2 lakh Dalton. And this complex have two cytochromes. Its names are A and A3. These are its features, this complex features. And it catalyzes this complex catalyzes transfer of electrons, that is transferred by or carried by the reduced cytochrome C to oxygen to release two molecules of water. Okay, and along with this process, here also translocation of protons taking place. And we are moving to the final phase, that is the generation or release of ATP, and that created gradient of what? Gradient means this. So this is the, sorry, mitochondria is not oval, perfect oval. Anyway, you just try to understand. Here itself, that gradient is generated, and because of this gradient, this proton can translocate downhill. Okay, downhill, and this transport occurs through another complex called the ATP synthase. Okay, because of this downhill transport, a certain amount of energy is released, and using that energy, the formed ATP here can release and can release or used up for various requirement inside the cell and outside the cell. Okay, that is the last step. And one more thing, from one NADH means if that electron transport chain starts from complex 1 using the electron carrier NADH, then how many ATP will produce? 2.5 ATP. What's the number? 2.5 ATP will form if NADH is the electron carrier. Okay, that means that chain starts from complex 1 onwards. That's the importance. But if the electron carrier is FADH2, then we know that the chain starts from complex 2. So we can produce, or that mitochondria can generate only 1.5 ATP in one transport pathway. Okay, that is the output. Next is about that ATP synthase. It's a main complex, enzyme complex that is generating the ATP. This ATP synthase is otherwise called F1F0 complex. You have to be familiar with that term also. Sometimes question can ask like that. F1F0 complex. It is a knob-like process, and it is also called elementary particle. Other name, its another name is elementary particle, or otherwise called oxysome. So be familiar with all these terms. In any of the way, there will be question for the exam. It's very important, ATP synthase, and it is synthesize the ATP. And in one mitochondria, there is about 15,000 different ATP syntheses. Then we can look to its structure. What are its component parts? F1, I will show that this is the F1, this portion, and F0 is the portion that is seen attached with the inner membrane, and this F1 is seen in the matrix. This F1 region is called it is having a head and a stalk. Okay, and this head and stalk composed of 5 different kinds of proteins. How many different? 5 different kinds of proteins from this head and stalk. Which are they? They are alpha, beta, gamma, delta, and eta. These are the different types of that proteins that form the head and stalk. And what is the ratio? And the alpha and beta, there is number is 3 each, and all the other is 1 each in this ATP synthase. This is the ratio. And F0 is made of the subunits called A, B, and C. And what's the ratio? In the case of bacteria, A subunit's number is only 1, and B subunit may present. And regarding C subunit, around 10 C subunit can present. And in the case of mammals, the number be same for A and B, but in the case of C, it's only 8. And this we understand that the number of C is more. So this C subunit itself forming the this rotating ring. This is a ring and having a channel means a hole, a narrow channel is present through this F0's rotating barrel shaped structure, and through that channel that proton is moving downhill to the matrix. Well, that's about the structure of the ATP synthase. And also this ATP synthase is called reversible coupling device. Why is it called so? Because we understand that we have learned that the ATP synthase generate ATP from ADP. In the same way, it can break down ATP to generate ADP. So that's why it's called a reversible coupling device. And the structure of this ATP synthase and its mechanism of mechanism of synthesizing ATP is explained by 2 scientists. They are Paul Boyer and John Walker. So with this we can finish today's class and section. Thank you.